Okay, this video is a review video for the on-level biology protein synthesis and mutations test. So we're going to start off with some vocabulary terms that you need to know for your upcoming test. The first one is DNA. So DNA stands for deoxyribonucleic acid, and it is in the shape of a double helix, and it's made up of nucleotides, and it holds the information for making any protein that your body might need to make. The next term is mRNA. mRNA stands for messenger RNA. It is single-stranded rather than double-stranded. It is also made of nucleotides and carries information for making proteins from the nucleus to the cytoplasm. It has groups of 3 bases on it that tell tRNA what order to bring amino acids in. tRNA stands for transfer RNA. Transfer RNA carries amino acids from the cytoplasm to the ribosome and matches its anticodon to the complementary codon on the mRNA, and together the tRNA and mRNA make sure that the amino acids in a protein end up in the correct sequence. Ribosomal RNA is not going to be a particularly important vocabulary term for you to know other than ribosomal RNA is part of the structure of a ribosome. Transcription is the first part of protein synthesis, and it is the production of messenger RNA from DNA, and it always happens in the nucleus because DNA can't leave the nucleus, and that's to ensure that anytime the cell needed to make the proteins, that the DNA would be there, so they would always have the instructions. Translation is the second step in protein synthesis, and it is the production of proteins from the temporary message in messenger RNA, and it happens in the cytoplasm on the ribosomes. A ribosome is the organelle that links amino acids together to make a polypeptide, and eventually that polypeptide folds into a protein. A codon is a group of 3 nucleotide bases on a messenger RNA strand, and it codes for 1 amino acid. So every 3 nucleotides represents or tells the cell what order to bring a particular amino acid in. And the anticodon is a group of 3 nucleotides on tRNA, and anticodons are complementary to the codons on the messenger RNA. A polypeptide is a group of amino acids that are linked together, and they eventually fold into a protein. Remember, the prefix poly means many. A mutation is any change in the nucleotide sequence of DNA. It is actually a change in the nitrogen base sequence. And a mutagen is any environmental factor that can cause a mutation. There are chemicals that can cause mutations. Ultraviolet light causes mutations. Sometimes mutations just happen by chance, though. So now we're going to compare and contrast DNA and RNA. DNA is always located in the nucleus, and RNA is made in the nucleus but travels to the cytoplasm. That's really the whole point of RNA, is to bring the code or the message from the DNA out of the nucleus so that the DNA can stay there, so that next time it's needed, you still have the information to make the next protein. The monomer of both DNA and RNA is a nucleotide. 3 of the nitrogen bases in DNA are the same as the nitrogen bases in RNA. Both of them have A, C, and G, adenine, cytosine, and guanine, but DNA has thymine, whereas RNA has uracil. And you can see the 2 bases that are different in DNA and RNA because I have made them purple on the screen. DNA is in the shape of a twisted ladder called a double helix. Now there are 3 types of RNA, and they vary in shape, but they are always single-stranded, so that's what the SS means on that particular screen. And in terms of size, DNA molecules are very large, and RNA is much smaller. Now, certainly RNA is a big molecule compared to many molecules, but if you're comparing DNA and RNA, DNA is much bigger because DNA is the instructions to make any protein that the body needs. You have 46 chromosomes, and a chromosome is a big piece of DNA, and you have more than 46 proteins that your body needs to make. Well, RNA, and if we're talking about messenger RNA here, it is a temporary copy of instructions to make one protein, so it's going to be much shorter than an entire chromosome of DNA because it's like a small piece. So what is the process that we're showing in this diagram? Well, you can see that there are two strands of DNA and it's open. So some students might say that looks kind of like a replication fork. However, one thing that you notice is that there's uracils in this new strand of nucleic acid. And if you see a nucleic acid that has uracil, you know that it's RNA. So if we're going from DNA to RNA, we have transcription. So the process in this diagram is transcription, not replication. Transcription uses genetic information on the template strand of DNA to synthesize a strand of complementary mRNA, and a short section of DNA that's used to make a strand of mRNA and codes for one single protein is called a gene. Transcription occurs in the nucleus of the cell, and the enzyme that separates the DNA and allows the messenger RNA to be built is called DNA helicase. It's the same enzyme that opens a DNA molecule during replication. Each sequence of 3 mRNA bases is called a codon, and it represents or designates one amino acid. There are 20 possible amino acids and 64 possible codons. You don't really need to memorize those numbers, although if you were going to memorize one of those two of those numbers, knowing the number of different types of amino acids is the more important number. AUG is the start codon for mRNA, and this codes for methionine. So methionine is the first amino acid in a protein. Remember, you guys watched a movie where they said that a good way to remember that AUG is the start codon is because it looks like the beginning of the word August, and school starts for you guys in August. In this diagram, we have translation, and we're going to be labeling the parts. So, what is that red strand? That would be mRNA. This structure, that would be tRNA. Now, it's not in this picture, but on this end there would be an amino acid, a very specific amino acid, and on this end there would be an anticodon. That organelle is the ribosome. That long strand of purple beads in this diagram is representing a polypeptide chain. And those individual monomers, the little purple spheres, those are amino acids. Proteins are polymers made up of monomers called amino acids that are linked together in peptide bonds. And remember, there's an enzyme in the ribosome that links them together. And if we're going to describe the role of tRNA in protein synthesis, tRNA ensures that the amino acid sequence is in the correct order. There's one tRNA for each amino acid, and the amino acid is on one end of the tRNA, and an anticodon is on the other end. The anticodon matches up to a specific complementary codon on the mRNA. Since mRNA carries the instructions for making a protein, when a tRNA anticodon and mRNA codon complementary base pair, the ribosome joins each amino acid that comes in the correct order. In the first step of translation, the start codon, AUG, attaches to the ribosome to begin the process of protein synthesis. Next, the amino acids are carried to the ribosome by tRNA molecules and are linked together. In the last stage, one of the 3 stop codons terminate the process of protein synthesis. It is not necessary to memorize the start or stop codons. You will always have a codon chart while taking a test. A mutation is a change in a nucleotide sequence of DNA, and mutations most directly affect the sequence of DNA nucleotides. So you have a different order of nitrogen bases in your DNA molecule. So the A's, T's, G's, and C's are now in a different sequence. So here we have 3 examples of a mutation And if you look at this diagram, you see the normal DNA sequence and the mutated sequence, and they're asking you to identify whether it is a point mutation, an insertion, or a deletion. So in this particular example, we have AAGCTG, and now we have AACTG. So what type of mutation do we have? We have a deletion because in this we are missing this guanine in the new mutated piece of DNA. So we are missing a base. So a base has been deleted. In example B, we go from CATGCA to CATACA. So we have 6 bases here and 6 bases here. So we do not have an insertion or deletion. One of our bases is now different. And in that case, we call it a point mutation because a G has been replaced by an A. And in example C, we have TCCGAT, and now we have TCCCGAT. So now we have an extra cytosine in here. So we have a new extra nitrogen base, so that's an insertion. So now it's asking you to circle the letter of the above mutations that would also be frameshift mutations. So if you have any extra base or missing base, that is going to cause a frameshift mutation. So both the insertion and deletion cause a change in the reading frame when the mRNA is being translated. So sometimes point mutations have no effect on a protein being produced. How can this be? Well, more than one codon codes for the same amino acid. Remember, on the previous slide, we said there are 20 amino acids, but 64 possible codons. So sometimes, even if one nucleotide is changed, it could still code for the same amino acid and make no change to the protein at all. And that is called a silent mutation. Why are insertions or deletions usually more disastrous than point mutations? Because all of the codons after the insertion or deletion would be changed due to the frameshift. So all the amino acids after that insertion or deletion would be different. So this makes the protein very different in shape and it won't function properly. So what effect would an insertion or deletion have on a protein? It would change many amino acids in the amino acid sequence, and it also could change a different codon into a stop codon and could make the protein much shorter. So what are 3 causes of DNA mutations? You can have random mutations because of environmental factors like chemicals or ultraviolet radiation. Sometimes there's no obvious cause and it's just an error made during DNA replication. Every time you copy DNA, there is a chance that a mistake will be made. And some causes of mutation would be inherited mutations like the BRCA2 gene, which makes women much more likely to get breast cancer if they inherit it. So now I'm going to remind you about how to use a codon chart. So in this slide, we've given you a sequence of bases of a potential messenger RNA molecule. So along the left side of your codon chart would be your first base. Across the top would be your second base. So if we have the codon AUG, we go to the row for A, and we know we're in this section of the codon chart. And U, we would be in this column, so we know we're in this square somewhere. So AUG would be methionine. Now for UAC, we're in this row, this column, so UAC would be tyrosine. UGU would be cysteine, and GGG would be glycine. So on your review sheet, we gave you a strand of DNA and you had to make the complementary DNA strand. And when you're doing that, A is always complementary to T and G is always complementary to C. And then we asked you to make a complementary messenger RNA strand. So T is complementary to A, A would be complementary now to U, because remember, RNA has no thymine, it has uracil instead. C would be complementary to G, and G would be complementary to C, and so on. And then if we use the codon chart from the previous page, we would get methionine, proline, and cysteine. And eventually we would come to our stop codon. So these are the answers to the last few questions of your test review sheet.